But without further ado, I'd like to introduce our first speaker, Sohini Ramachandran, and she's been multiple year CGSI speaker and one of our best rated, best reviewed speakers. We're really excited to hear about what she's going to say. So Sohini, feel free to share your screen and get started.
Thanks a lot, Eleazar, and well, thank you for that kind introduction. And I've always loved being a part of CGSI. It's nice to see so many familiar faces. So today I wanted to tell you about largely unpublished work, just kind of new provocative threads from my group. I'm a population geneticist, for those of you who don't know me, but increasingly becoming interested in how population genetics and statistical genetics should intersect more and more as we try to study complex trait architecture in humans. And I'm very inspired by the work of many people who are part of the CGSI community, especially Bogdan Pasanuk and John Novembre. So it's nice to see you all here too. My career to date, I think, has been very inspired, like many of us perhaps, by the fact that our own species has a really interesting and recently shared dynamic evolutionary history. So our modern human evolutionary history features a lot of complex dynamics that have happened in a relatively recent amount of evolutionary time, spreading through multiple habitats rapidly, experiencing changes in diets and pathogens. And that's what really shapes our genomic diversity. And in order to identify the genetic targets of natural selection, we need to condition on this history somehow. So when we try as population geneticists to study the role of, say, adaptation genetically or disease-causing mutations in shaping genetic variation, we really need to think about population histories when we do that. So what I want to talk to you about today, and I know this will be familiar to most everyone on the call, but I just want to be sort of overly pedantic to make sure we're all on the same page, is how this complex evolutionary history that our species shares interacts with genome-wide association studies of disease, which I view as a hypothesis-generating approach to study complex trait architecture, where we use marginal associations at different variants or SNPs throughout the genome with phenotype states. So what this looks like in practice is that for every variant genome-wide, we sort of fit a particular model, usually this kind of linear model, where we're regressing a phenotype state, say height, onto genotype state, which could be coded many ways, including as dosage of a particular allele, say 0, 1, or 2 at a particular variant site. And the overall model for GWAS is quite simple. It's really this linear regression where we might have, phenotype here as our dependent variable, this allelic dosage term here as an independent variable, and then other covariates that could be related phenotypes often used, and then other covariates used to control for population structure. But overall, our interest is in this slope parameter here and whether or not statistically different from 0. So another way of thinking about the model for GWAS is that it's really setting up a classification problem. And the problem is to see whether a variant directly influences the trait. I'm going to use influence very broadly, but I'll say we want to call these causal SNPs. These could also be tagging perfectly a direct influencer on a trait, or to be non-causal. And as a field, we haven't really broadly adopted methods that actually get at causality, like causal inference techniques, but this is sort of, one interpretive goal of GWAS. So then, again, many of you will be familiar with this, but what happens is we do this genome-wide, correct for multiple tests, and then usually pursue different functional validation techniques for these hypotheses that have been generated. And I think when thinking about GWAS, it's really interesting to return to the very first GWAS. So this was the GWAS on age-related macular degeneration, published by Klein et al. In Science in 2005. many of you on the call may have been like me in graduate school when this happened. So this was one of the first Manhattan plots. And what I think is interesting to remember about this study is that there were 96 cases and 50 controls. And GWAS was effective here in that it identified mutations of large effect that underlie age-related macular degeneration, particularly an intronic and common variant in complement factor H that was strongly associated with this disease. So I think, many of us who develop methods know that the biggest challenge of GWAS is really the data generation, and the data curation. The model itself is quite simple, and the standard vanilla model. So going back to the classification problem, another way of viewing this is to think about a very particular model, that there are sort of causal SNPs emitting effect sizes that are probably normally distributed, or we assume that's the case, and that this component has kind of a wide variance. And the effect sizes of non-causal SNPs are assumed to be 0. So you could depict these as this point mass here at 0. And this idea, this is a very strict null model, and the consequences of it with respect to false discovery rate have been popularized, I would say, recently by Matthew Stevens in our field, but were formally introduced by Brad Efron around 2007 in statistics. And what's really interesting about the strictness of this model is that under this null, if we have enough samples, and I would argue that we're sort of there as a field and have been for a few years, we end up in a world where everything is statistically associated with a trait of interest. And we've seen that happen in GWAS over time, that now the proportion of phenotypic variants explained by individual SNPs being identified in larger and larger studies is getting smaller and smaller. We've achieved diminishing returns, and I think it's an interesting time to question what we're sort of learning biologically from statistically significant variants in GWAS. I think, another interesting feature of GWAS, again, is that only very recently is it starting to be interpreted through the lens of population genetics. And one way that that's being done is by talking about the study design, which is very, very skewed and doesn't represent the diversity of human genomes throughout the world. This has been depicted in many ways over the years, starting as early as 2007. But this is the most recent graphic that I find to be incredibly compelling by Alicia Martin, published in Nature Genetics in a perspective piece last year. So this shows the cumulative distribution since the first GWASs up until now of ancestry of samples used in studies. And you can see that individuals of European ancestry make up the vast majority of individuals being analyzed in GWASs, quite disproportionate to their representation in our world. So across over 4,500 genome-wide association studies, about 90% of the samples represent just 16% of the human population. And there have been lots of questions about what that means for what we can extrapolate from GWASs. More recently, Simone Gravel led a group effort to examine the extent to which genetic data from emerging large-scale biobanks is being used in order to rectify this study design problem. And what he found, this group of researchers found in a comment in Nature in the fall last year, was that even in large, diverse biobanks where the sample sizes of non-white individuals are larger than we might have ever hoped for a few years ago in GWASs, those groups are being ignored. And he looks at this particularly in the UK Biobank and also in one other US sample. I'm sorry, it's escaping my brain at this moment. But my group, we did a PubMed scraping study where we looked at all abstracts in the last few years that have mentioned the UK Biobank and cross-referenced those with studies that were listed on the UK Biobank website. But basically, we're trying to identify PubMed IDs of papers that analyze UK Biobank. And what we find is that when we look at those studies with metadata in the GWAS catalog, essentially they are all analyzing what's called the white British cohort in the UK Biobank, which is almost 400,000 individuals. But the UK Biobank is a database of 500,000 residents of the UK. And it includes a number of South Asian ancestry individuals, African ancestry individuals that are essentially not being studied despite the large vastness of this resource. Okay, but what else could we do kind of as methods developers to think about how population genetics and statistical genetics might connect? Well, I think another important thing for us to remember is that we know from a number of studies of large data sets assaying human genetic variation that linkage disequilibrium, which I'll define in a minute, increases quite systematically with genetic divergence from Africa. And this complicates localizing both adaptive and deleterious mutations. This was first depicted by Noah Rosenberg's group, led in an analysis by Matthias Jakobsen in Nature in 2008. What this is showing is linkage disequilibrium, or as Eleazar has said multiple times at CGSI in very compelling talks, it's just correlation. And there are lots of great reasons for it to be just correlation, but Pearson's correlation in genotype state at a fixed window size. So this increases really systematically with a proxy for genetic divergence from Africa, meaning that the African genomes have had more time to break up associations between genotypes due to recombination events than have other ancestries. And the longest shared sequences amongst individuals are observed in indigenous American populations. And this has been recapitulated many times by work that I've been involved with, but also many other groups. Okay, so how do we incorporate this idea into something like this is what I want to talk to you. So leveraging the diversity of data that's in some of these biobanks involve variations on a theme of view. So here are some thoughts that I just want to kind of pose to you, and then I'll show you some sort of preliminary results that I'm not gonna sort of hang my hat on, but just again, to propose some provocative questions for you this morning or afternoon, depending on where you are. So one question is whether or not causal variants are spread widely across the genome or clumped into biologically relevant genes and pathways. This is a question I've been quite interested in for a while, and it was specifically posed by many people, but also in the paper that posed the omnigenic model by Jonathan Pritchard's group in 2017. The work I've done in this area has to do with developing gene level association tests, one of which was published in 2016 in Genetics, and another that came out this year that I'll talk a little bit about, or I'll mention. Another question is, to what extent do we expect the genetic architectures of human complex traits to potentially vary by ancestry? We might not expect that to be the case for a trait that's closely tied to fitness, for example, but we might expect that there are ancestry-specific modifiers, genetic modifiers of architecture, and how might we develop methods to think about that? Are the same regions enriched for contributions to narrow sense heritability across ancestries? Are a lot of our approaches that have looked at how the phenotypic variance is explained across the genome have only recently started to get applied in a multi-ethnic context. And I think it would be interesting to talk about how to interpret different results we might see when we do that. And lastly, I'm not going to touch on this at all, but I think for those of you who might be interested in phenotypic prediction, one question is how well we can predict out of sample. So if we're doing a lot of our studies in training and testing cases that aren't representative of other studies we might like to do downstream, how exactly do these results transfer? So what I'm going to tell you about is in a paper that we haven't yet posted as a preprint, but I'm hoping we will by the end of this month. So keep an eye out for this. It's led by a wonderful grad student of mine named Sam Smith. We'll be looking for postdocs soon. Okay, so what I want to pose to you is, what if we move beyond the variant in GWAS studies in multi-ethnic groups when we're trying to think about complex trait architecture? So another way of thinking about this is, given that LD differs across ancestries, should we really expect mutation level associations to replicate in multi-ethnic GWAS studies? So one way we could think about, this is the current null hypothesis in a multi-ethnic GWAS study, is that mutation level effects are the same across ancestries. So but we could think about a series of kind of nested models that are actually the ground truth architecture for different traits, because there are exceptions to this idea that the mutation level architecture is the same across groups. So there are many traits that seem to adhere to this model. Inflammatory bowel diseases are one. But you could also imagine another kind of model where, say, the core genes that are perturbed in a trait are the same across all ancestries, but the causal mutations could be different. And I think this is the motivating case for a lot of gene level association studies, and also studies that try to map GWAS data to EQTLs, for example. One example is ApoE4 alleles and how they have differential effects in different ancestries. I also want to point out that there are many Mendelian diseases that follow this kind of model. So even a monogenic disease doesn't necessarily have just one shared mutation that gets perturbed in all cases. You could imagine that core genes might differ across ancestries, but they all are biologically interacting, perhaps in the same pathway. Skin pigmentation is one example of this, as demonstrated by Brenna Henn's group. And then just for completeness, we could imagine, the complement of these three cases, which is maybe less biologically believable, but perhaps there could even be very different ancestry-based genetic mechanisms for the same trait. So really quickly, in the interest of time, I'll just show you something that is intuitive probably for many of you, is that if we aggregate SNP level association results in, say, biologically relevant units like genes or pathways, we can actually come up with really targeted hypotheses about trait architecture. So I'm going to show you power in simulation to detect ground truth causal mutations or genes containing ground truth causal mutations in a series of methods. And this is across a series of architectures. So here across the x-axis, sample sizes are changing, as well as genetic heritability of the trait. So the higher numbers mean the trait is more governed by genetics versus the lower numbers. Okay, so here's what GWAS power looks like. It's well powered. The more samples we have, the better we do. The stronger the genetic basis for the trait, the better we do. Here is the method Genie that we published in 2020 here in Cheng et al. And PLOS Genetics. It also does pretty well, maybe slightly outcompetes GWAS for a highly heritable trait. And there are related models that you might be familiar with, RSS from Matthew Stevens' group and the SCAT family of tests that also do sort of quite well. But, looking at this, you could say, well, these are all sort of well powered. But let's look now at false discovery rate. So this is really where, again, we see challenges of GWAS. So GWAS is calling lots of things positive, and when true positives, or classifying many things as associated. So when you do that, you end up with a lot of false discoveries as well. And this is what gene level association tests can control really well for. So just on the methodological side, I'll say kind of briefly that I would say these methods highlight in order their benefits of aggregating SNP level association statistics, benefits of doing shrinkage due to inflated GWAS effect sizes, and then benefits of modeling LD on top of that. And I think that's what this plot is illustrating. So what if we apply some of these gene or pathway level association tests at different genomic scales across ancestry? So I'll just leave you with one last set of results from this study where we're looking at UK Biobank, Biobank Japan, and the PAGE Consortium data. We have a large range of sample sizes in the third column across these ancestries, but many of them have been assayed for a similar set of quantitative traits and biomarkers. Okay, so what we've done here is take Genie, apply it to genes throughout the genome, and then assess whether those genes are interacting with each other using a method called HotNet from Ben Raphael's group to try to identify strongly connected gene subnetworks that are perturbed in cases, in this case of triglyceride levels. When we look at European ancestry individuals, we identify a cluster of apolipoproteins known to biologically influence triglyceride levels that are all interacting with each other and all perturbed or jointly perturbed in many cases. We see a lot of overlap in this subnetwork when we look at Biobank Japan, as well as sort of a new association enters the ring, and this gene base 1 that has been identified as associated with triglyceride levels in GWASs of Asian ancestry individuals. But even when we look at the pretty small native Hawaiian cohort, I apologize that this is Oceanian, in the PAGE study data, which is a quite small sample size, we actually identify other genes enriched for mutations in cases that are biologically interacting with this subnetwork. So what I'm trying to illustrate here in this very qualitative result is the idea that I think with multi-ethnic data sets, it's a mistake to throw away any cohort. And in fact, one analogy we might use is to think about the blind men and the elephant, where no one sort of viewpoint is going to tell us what structure we're looking at, but maybe looking at each of them together is going to help us understand more about the biological basis of this trait. All right, so in the interest of time, I'm just going to skip over heritability and say that I think a challenge in multi-ethnic GWAS is that if we expand data sets, which we should definitely do, if we continue to do more of the same methodologically, particularly with respect to the traditional GWAS approach, I think that will be a mistake. And I think that a lot of people on this call are trying to work to create methods that will improve that situation. I do think that if we stop focusing on variants and try to think more about biologically interpretable units in the genome, that we will generate better hypotheses for understanding genetic trait architecture, even in the face of variable sample sizes. I'd also be intrigued to talk with more folks here about ways that we might in our field try to unify prediction and association, which I think in principle we should be able to unify, but we've sort of taken different directions for answering those problems. And lastly, I would argue that bringing an evolutionary perspective to biomedical research, in particular to medical genetics, is sort of crucial, especially for making sure personalized medicine is a reality for everyone. So lastly, I'll just mention that this was all work done by my group and in collaboration with Ben Raphael, another CGSI faculty member. And I'm also looking for new postdocs, so get in touch if you're interested or know someone who is. Thanks.
Great, Sohini. Thank you for the wonderful talk. So now we take questions. So either speak up or leave a question in the chat. And we have about 5 minutes for questions. Really great talk.
So I have a question. This is Bogdan speaking. Fascinating talk, Sohini. Really exciting. Just curious whether you can comment a little bit from the evolutionary perspective. So we all know that LD as well as frequencies vary by ancestries, and that's a big source of heterogeneity by ancestry. I'm just curious whether you have insights, particularly from the evolutionary perspective, of allelic effects varying a lot by ancestry or not, or in general, what's the interplay between changes in 1 unit of the substitution by ancestry versus frequency changing. And I'm thinking more in terms of how selection would have impacted those.
Yeah, no, it's a great question, and not one that I have explicitly worked on, but I would say, I mean, as you know, like I think Kirk Glomuller is doing some really exciting work in this space, and I think that his work with his student Arun Dorvasula is really interesting to think about how, a trait being tied to fitness might lead to this situation. I think Doc Edge has also done some great work on gene by environment effects and how these might manifest as well. So I'll just say, I'm kind of a groupie of those sorts of methods, but yeah, I think it's a great question. So both whether, say, from a transferability standpoint, the actual SNPs transfer, but also the extent to which the effect sizes do. And that's going to be the extent of my comment right now for you. I apologize. But right, I think there's a lot of work to be done in both directions.
Hi, Sohini. Great talk.
Sure.
Thank you. So I had a question. So did you do any formal, you listed these the different models. So did you do any kind of formal model comparisons to see whether they explain the data that you talked about presented on ApoE?
Yeah, so that, I mean, again, I apologize for due to the short amount of time, I couldn't really get into it, but we do in the preprint. And so I'll say that part of what we're trying to demonstrate there as well is that, so GWAS on its own doesn't identify that whole substructure, even in European ancestries. Other models do, other gene level association tests. One issue is computational scalability, if that's the question that you're asking. Sorry, is it a methods comparison question? Yeah, so for example, RSS and Genie have very, very similar models, but since RSS is fully Bayesian, it's really hard to get it to scale to genome-wide comparison. So, so we talk about that in the paper.
Sohini, this is John. I'm wondering about, I found very provocative the part at the end about the different pathways and this analogy of, the different parts of the elephant and the Oceania causal genes being part of the same network, but a different sort of corner of the network, right? And I'm wondering if we might, be able to look at sort of null models of demographic history and to what extent you'd expect it to partition in certain, certain ways. I imagine it's a complex question of like, if the rate of mutation is low, then it might be very, like a population will only have sampled one part of the space. And so there might be more partitioning, but at a certain high level of mutation, every population should have sampled every gene within a pathway. And then maybe invoking other forces like selection, if there's some pleiotropic effect of some of the genes on something else, that might lead that one population, because of local adaptation, have explored that one pathway. But the first two of those could be explored with sort of neutral models of mutation or sort of in a way that might be interesting. I don't know if that's...
Yeah, no, that's in fact exactly what a couple of my students are interested in right now. And I am as well. So I'll just say, like, right now, I'm not, and not that you're saying this, I'm not invoking, say, ancestry-specific genetic architecture to explain that. I'm just intrigued by the idea that if we sort of look at different genomic scales and in different ancestries, we might identify things that are different from the perspective of statistical significance, but biologically sort of related, right? And maybe have similar biological significance. But I think, yeah, so exploring the extent to which we might expect to see such a partitioning with different levels of FST, say in admixed individuals, for example, between ancestry sources is something that we're trying to model right now and think about. It's a little complicated because as much as I am a fan of aggregating these SNP level effects, there is also this fundamental challenge that many of you will be familiar with from your own work of like how to map a SNP to a gene or a region is also complicated. So there's also that question of, perturbation. And then we all are organisms with many phenotypes, right? So then if you have, so I think there are lots of interesting questions to think about in the multivariate sense as well. Like what if one phenotype is under selection, strong selection, but is also related to architecturally another phenotype that maybe isn't, like what happens with all of these models. So I think there's a lot of work to be done around methods and asking exactly what you're saying of to what extent do we expect multi-ethnic data sets to help us gain insight into a fixed genetic architecture trait.
Okay, thank you so much, Sohini, for the great talk and the great discussion. Really enjoyed it.